Hi everybody, welcome back to another video. In today's video, we're going to talk about skeletal muscle mechanics, motor unit recruitment, what they are, and force production. And at the end, we'll talk a little bit about fiber type distribution in skeletal muscles of athletes and non-athletes and adaptations to exercise. Okay, so without any further ado, let's get started. If you enjoy the video, please like, please comment, please subscribe. It'll mean a lot to me. And let's get going. So first things first is, so far we've been looking at, a generalized view of skeletal muscle mechanics, right? We've just shown one neuron, right, coming in and innervating this muscle fiber, right? And we've seen how that muscle fiber will contract in response to excitation, okay? This view is rather simplified because we have multiple motor neurons innervating a whole muscle, and we have multiple different types of muscle fibers within a muscle. Okay, so we're going to take a look at these, stuff today in deeper detail. But first things first, we've seen this image. Okay, we've seen this image in the previous video, but now I've gone ahead and expanded it a little bit. Where again, we're seeing this motor input coming down from the higher brain centers to different levels of the spinal cord. Okay, different segments of the spinal cord, if you will, and they're going to synapse on these motor neurons, and these motor neurons, of course, are going to go out and innervate different fibers within a muscle fiber, within a muscle entirely. So this right here, I'm going to write down, is a single muscle, okay, a whole single muscle. We're going to say this is, for example, the biceps, okay? And we're going to say on this side, this is another single muscle, and we'll call it, for example, the triceps, okay? Now, this entire single muscle, right, has a whole bunch of muscle fibers in it, right? And it has different types of muscle fibers inside of that single muscle. Now, each one of those different types of fibers is innervated by a different motor neuron, all right? So we'll see later in this video, we have different kinds of fibers. We have type 1 fibers, we have type 2A fibers, and we have type 2X fibers. Each one of these fibers is innervated by a separate motor neuron. So here's a motor neuron innervating that guy, a motor neuron innervating that guy, and a motor neuron innervating that guy. All of the motor neurons that innervate muscle fibers in a single muscle lie in the exact same spinal cord segment, and they are referred to as a motor neuron pool. Okay, so this guy is a motor neuron pool for this muscle. These are all of the individual motor neurons innervating the fibers in this single muscle. Okay, now these guys can extend anywhere between 1 to 4 spinal segments. I've only got drawn 3 segments here. Okay, we can have this example here. We can have another muscle such as the triceps. It has its own set of muscle fibers, right? And its own different distribution of muscle fibers in it. And you got motor neurons innervating those fibers, right? Again, all of the individual motor neurons innervating a single muscle, in this case the triceps, are going to be lying in a separate motor neuron pool. So here's a motor neuron pool number 2. This is number 1, okay? And these guys, we're going to go and innervate that, okay? All right, so just want to get that point across there. Now, let's go ahead and speak about these motor neurons and their muscle fibers a bit closely. So we said that we have different types of muscle fibers, right? So we're going to have type 1 fibers, we're going to have type 2A fibers, we're going to have type 2X fibers, right? And these different kinds of muscle fibers have different contractile and metabolic properties, and they also have different motor neurons, all right, as we were just discussing. So a type 1 motor neuron looks like this, okay? And a type 2A motor neuron looks like this, and a type 2X looks like this. Let's, for just a second, look at this image. Let's just look at this image, and let's start, finding differences. What are some of the differences you see? The difference I see here is, 1, the type 1 motor neuron is smaller compared to the type 2 motor neurons, okay? What else? I see that it branches out less compared to the other 2. I see that it has a smaller surface area and a smaller diameter here as compared to these guys. And that's all I see for now. Another thing I see is that the type 1 fibers, so if I orient you proper, this is a motor neuron, right? The alpha motor neuron. And this is the myofiber. And since this is a type 1, this is a type 1 myofiber. What differences do you see between this myofiber and all the other myofibers drawn here? Well, a big glaring difference is their size. The type 1 myofiber seems to be smaller than the type 2 myofibers. Okay, what else? I see its color. Its color seems to be more of a dark red, and the other two seem to be a bit of a lighter shade of red, and this one's more of like a pale pink. Okay, so colors are different there. I see the number of them is different. Okay, I see that this type 1, this motor neuron that's innervating these type 1 fibers, is innervating a fewer number of these myofibers compared to the type 2 motor neurons. They're innervating more. Okay, all of these differences that we're pointing out here, they're not by mistake. This is all accurate, all right? The number of them, sure, isn't accurate, but the pattern is completely accurate. A type 1 motor neuron is smaller than the type 2s. And it does branch out less because it is innervating a lower number of type 1 myofibers. And if we were to compare that to a type 2, you can see that the type 2 is not only bigger in diameter and surface area, but its branching is even more extensive, and it's innervating type 2X fibers, okay? All right, we'll dig into these more, believe me. Some other things that you should remember from neurology is that the axon's diameter is, proportional to its conduction velocity. So this axon has a small diameter, so it must have a slow conduction velocity, okay? This axon, okay, it's in the middle of the two, so we're going to say intermediate conduction. This axon is the biggest, so it has the fastest conduction. Okay? All right. Next is something else. It is the recruitability of these neurons. Remember, these green guys are these green neurons here, right? They sit in the ventral gray horn of the spinal cord, and they're waiting from upper brain center inputs to get them activated. And then they're going to send action potentials down to the muscle, right? Recruitability of these neurons is a little different as well. The surface area for this dude is real small, okay? So imagine if I have two synaptic inputs, and two synaptic inputs, okay? Now imagine both of these synaptic inputs are going to cause the exact same level of depolarization on each cell body here, okay? Meaning that this guy will, for example, cause a 50 millivolt change. This guy will cause an exact 50 millivolt change. This guy will cause an exact 50 millivolt change. However, there is a big difference here. These smaller motor neurons, they have a smaller surface area. This means they're going to reach threshold much sooner. And these guys, because they have a larger surface area, they're going to reach threshold much later. So the exact same synaptic input that barely depolarizes this large neuron completely brings this first smaller neuron to threshold, okay? And so recruitment of these guys is different as well, all right? This guy, because he's smaller, with a smaller amount of synaptic input, will be able to be easily recruited. This guy, intermediate. This guy, the hardest to recruit. Why? Because of the shape of this actual neuron here, okay? All right. Next is that this recruitment order we'll come back to, okay? It is important, and we'll come back to it. But next thing is the force production level. Remember we were talking about the shape here, right? We said that this guy is smaller. Well, remember something else? In the very first video, I said that a myofiber, right? 80% of the cell volume of a myofiber is what? A myofibril. So myofibrils make up 80% of the cell volume of a myofiber, and guess what? That 80% will be quite small in this small fiber, but 80% of this big fiber is a lot of myofibrils. And so just purely off of the virtue that these type 2X fibers have a bigger diameter, they can fit more myofibrils inside them as compared to smaller fibers. Why does it matter if I can do that? Well, myofibrils have sarcomeres in them, and sarcomeres are the smallest functional unit, and force production arises from the sarcomere. So if we follow the math, type 2X fibers, because they have more myofibrils, can produce higher force as compared to these guys. Okay? All right. So force production is also different between these guys. Many other things are different, as we'll come to see. We'll come to see that type 1 fibers are fatigue resistant, meaning that they can maintain force production over long periods of time without their maximum amount of force production dying down. So if we were to look at this on a graph, here is fatigue curve. On the y-axis, I have the maximum, the percentage of my maximum force production over time in minutes. And in a type 1 fiber, you'll see that it will be able to consistently meet 100% of its maximal force production over time. And you will see that by 60 minutes, this fiber type is still producing the exact same level of force that it was at 1 minute. What does that tell you? This is fatigue resistant. We'll come to see why. Type 2 fibers, not so fatigue resistant, okay? They're not so fatigue resistant. And we can see an example of this that, for example, this type 2A fiber, it starts off at 100%, right? Percentage of maximal force, remember? It starts out at 100%, and then by the 2-minute mark, right, it will start going down, okay? And by the 60-minute mark, you can see that it's only got a small amount of its maximum force production still running up. We can compare this type 2A to the type 2X to really understand why this middle fiber is always kind of intermediate between the two. If you were to take a look at the fatigue curve of a type 2X fiber, you will see that it immediately reaches 100% of its force production really fast, and it drastically dies down within the first minute, and it becomes really, really sparse in force production by 60 minutes, okay? This is important because this goes to show you that this fiber type is really used for powerful bursts of force production rather than longevity of a contraction, whereas this is more so used for longevity of a contraction. Again, this all has to do with their metabolic properties that differ. They're all fiber types. They all contract the exact same way, but their metabolic properties change things around, okay? We'll come to see that the speed of contraction is different as well. The type 2X fibers have the fastest speed of contraction. The type 2A fibers have an intermediate speed, slower than type 2X, faster than type 1. And type 1 fibers have the slowest contractile kinetics. We can draw another graph for this. Here's a graph for these guys. You can see that in a type 1 fiber, the duration of contraction is much longer, okay? So in the previous video, I introduced you guys a concept of a twitch, a muscle twitch that arises from a single action potential, right? These graphs represent a single twitch of these different fibers. And again, a twitch is a single bout of mechanical activity arising from one action potential. And so you can see not only is the force production minimal in one twitch from a type 1 fiber, but the period of contraction is quite high, all right? And in the last video, we said that it's around 100 milliseconds for a fiber. That's more of a generalized number. For a type 1 fiber, this would be around 80 to 120 milliseconds long, the entire mechanical activity. Whereas in a type 2X fiber, you see another bit of difference. You see that the force production has now gone up compared to type 1 just from a single twitch. And this should make sense because of this myofibril size. It's bigger than type 1, okay? But the other thing you see is that the twitch duration is smaller compared to this guy, okay? And so if the twitch duration is smaller, right, the type 2A would be around somewhere of 40 to 70 milliseconds, right? So the entire twitch for a type 2A lasts around 40 to 70 milliseconds. Again, these are ranges. And for the type 2X, you see the biggest increase in force, okay? And again, this tracks with the myofiber size. And you can see the slowest contractile kinetics in a single twitch. And this will be, for example, 20 to 40 milliseconds, okay? Again, we've taken a look at these fatigue curves, these force production and twitch kinetic curves, all right? These are important characteristics of these fibers to understand. And we'll dig into what actually creates these characteristics later. Okay, now I want to tell you guys more about the names of these fibers, okay? These fibers often go by different names in the literature. You can call them slow twitch. So this type 1 fiber can go by slow twitch red. And slow twitch is referring to its contractile kinetics, which we just saw is the slowest out of all three. And its color, it is red as opposed to the other guys, okay? It is also called slow oxidative, and this is referring to its main method of energy production, and we'll look at this later. Type 2A is deemed fast twitch red because it is still red, but it is now fast twitch because of its contractile kinetics, which again, we took a look at. Its other name is fast oxidative glycolytic, and this refers to its main method of energy production. We'll come to see that the type 2A fibers were always in the middle of type 1 and type 2X. They're not going to make up their mind almost, right? They're kind of in the middle always. They always got a best of both worlds. Type 2X is referred to as fast twitch white, and again, that has a reason. And if you can take a look at it, the fibers look pale in comparison to the other guys, and so they're fast twitch white. And this one will be deemed as fast glycolytic. Again, refers to its main energy production. So these are all the names that you should be familiar with for these fibers. Now, the other thing I want to introduce you to is this concept of orderly recruitment and the fact that we recruit fibers not just in a random fashion, but in the exact same fashion, in the exact same order every time. And there's a reason for that. But first, we have to talk about something. And that thing is the concept of a motor unit. So this entire thing is a motor unit, MU. Okay? What is a motor unit? A motor unit is defined as a motor neuron plus all of the myofibers that motor neuron is innervating. And to be more specific, it is a motor neuron and all of the same type of myofibers it is innervating. Why do I say same type? Because a motor neuron can never innervate different types of fibers. It will always be the same type. So this motor neuron, this small motor neuron, will always innervate these type 1 fibers. It cannot go ahead and innervate a type 2A fiber or a type 2X fiber. That doesn't make sense. Can you imagine this? A motor neuron gets activated and fires. And that motor neuron somehow in bizarre world is innervating a type 1 and a type 2 fiber. Force production would look quite weird. And the fatigability would then look quite weird as well, because these have different metabolic contractile properties. So it doesn't work. That's what a motor unit is. It is a motor neuron and the myofibers of the same type that it innervates. We can have different sizes of motor units. We can have large motor units, as evidenced here, and we can have small motor units, and here. And then there's, of course, the intermediate size motor units, which is these guys. Now, based off of everything that I've told you, the fatigability of these fibers, their contractile kinetics, their force production, the recruitability of the motor neurons, which of these, if you were to be designing a human and you wanted to make it so that it is the most efficient for this human to contract its muscle fiber, meaning the force production is going to be efficient, the energy usage is going to be efficient, and the fatigability is going to be efficient. What is the order of recruitment that you would put here? I'll tell you the order of recruitment that would make sense for me. It has to do with effort. Why would the central nervous system put a lot of effort, okay, to recruit these large motor units and produce a whole bunch of force for me just wanting to lift a water bottle, which isn't that heavy? Why would it want to do that? It doesn't make sense. What does make sense is for it to start off of the smaller motor units, and if we can't meet force production demands, gradually make your way up. You know what that allows? That allows for force gradation. We'll talk about this at the end of the video when we talk about how do we increase force. And this force gradation is important because I don't have to, as an organism, waste energy doing some trivial task that I could have done with minimal amounts of effort. And so I will start by recruiting a type 1 fiber, which is the easiest to recruit. And it has the most fatigue-resistant fibers. And even though they don't provide the highest amount of force, if I recruit multiple type 1 motor units, right, because we have multiple of these motor units inside of a muscle, then I will generate a lot of force. You know what that orderly recruitment is called? It is called Hahnemann's size principle. Hahnemann's size principle describes the principle that motor units are recruited from small to large based on their recruitability and their threshold. Remember, we said these guys have a low threshold, these guys have a high threshold, so they're harder to recruit, they require more effort, right? That's important. Effort is important here. So the motor units are recruited according to their magnitude of force output and their recruitability, all right? From smallest to largest. And I said this has multiple benefits. It allows for gradation of force, okay? It allows me to pick up a water bottle without using this type 2X fiber and forcefully picking it up and slamming it to my head. It allows me to perform fine movements by recruiting this small motor unit first. The smaller the motor unit, the finer the movement can be done. What do I mean from this? So let's take this example. You see this right here, the small motor unit. It's only innervating what? 2 fibers. We say that's an innervation ratio. Innervation ratio. And the smaller this enervation ratio is, the more level of control I have in a movement. Okay? What do I mean by this? For example, you know your extraocular muscles? These are your muscles that help move your eyes left, right, up, down. Do you want forceful amounts of force production in your extraocular muscles? No. You want fine control, precision movement. Okay? How do you get that? You get that by lowering the enervation ratio, because the smaller this enervation ratio is, less force production I have, and the more control of the movement I have. The higher this enervation ratio is, the less control of force I have. What's an example of this? Extraocular muscles have an enervation ratio of 1 to 23. That's one motor neuron enervating only 23 myofibers, and they're likely to be of type 1. Okay? And so this is good because extraocular muscles, I don't want them to produce a lot of force. I want them to be precisioned, all right? They don't need a lot of force. They work all day to, move my eyes around. They need to be fatigue resistant, and they need to not make a lot of force. I need to have control over them. My leg muscles, on the other hand, they can have much higher orders of magnitude. So 1 to 2,000 is what's observed in leg muscles quite commonly, okay? So for example, the quads. That means one motor neuron is enervating 2,000 myofibers. In this scenario, do I have a lot of control? No. Can I produce a lot of force though? Yes. So force production is high here, but fine motor control is not occurring. This is for fine motor control. This is for force production, okay? Where is that force production coming from? Well, it's coming from the fact that these fibers, these type 2X fibers, based off of their diameter, have more myofibrils, so they inherently generate more force. That's one. And two is that this motor neuron is enervating multiple types of these large fibers, and so the motor unit as a whole is going to generate a lot of force. Whereas the small motor unit is the exact opposite case. These fibers inherently don't produce a lot of force. They are inherently fatigue resistant though, and the motor neuron only branches out a small few times and enervates a small number of these small myofibers. And so force production as a motor unit will be smaller. Okay? These are important concepts to be able to understand. So with the Henneman size principle, these are the advantages. There are other advantages of this orderly recruitment, right? We're going to minimize fatigue, like I said, by first activating these fatigue-resistant type 1 muscle fibers, and then the larger, more powerful units are recruited whenever we need them, whenever the force demand is higher. We talked about the fine control of muscle forces. That is also good. That's another advantage. And finally, it just removes the burden of determining which motor neuron to activate first by the central nervous system. It just occurs automatically according to the size of the motor unit and its threshold. Okay? A great depiction of this Henneman size principle is in this graph right here, where on the x-axis we have the percentage of active muscle fibers, and on the y-axis we have the percentage of active muscle fibers, and on the x-axis we have exercise intensity. And so with increasing exercise intensity, the percentage of active muscle fibers increases. And I'll tell you this right here. This guy is my type 1 fiber. This line is my type 2A, and this is my type 2X. What the hell does this have to do with the Henneman size principle? At a given exercise intensity, let's just say at 0% of my VO2 max, okay, to around 40% of my VO2 max. This is just a level of exercise intensity. I am using my type 1 fibers. But then if I increase it past 40%, so now I go to, 60% of my VO2 max. Now what happens? In this 60% VO2 max, my type 1 fibers are no longer going to be able to provide the necessary force for me to maintain exercise at this 60% intensity level. And so the only thing left for me to do is recruit the next fiber type that will allow me to continue my exercise intensity at this level. And that is the type 2A fiber. The type 2A fiber will then help us maintain exercise at this 60% VO2 max. But notice that the type 1 fiber is here. It's just on top of it, we have now recruited type 2A. Same principle goes if we go up to, for example, 75% VO2 max or 80%. At 80% VO2 max, which again is just a measure of exercise intensity, my type 2A and type 1 fibers together can't help me meet this demand. I can't maintain my exercise intensity at this demand. I need to go to the next recruitment. I need to recruit type 2X fibers to help me meet force production to maintain exercise at this level. This occurs all the time. This is the Henneman size principle. This is a great depiction of the Henneman size principle. Okay? Please also understand that motor unit recruitment always occurs type 1, type 2A, then type 2X. You cannot change this order. Okay? This order will forever remain like this. There are instances in severe fatigue scenarios where this might change, but generally speaking, this recruitment order will always remain. If I start doing a 100-meter dash, I don't randomly just start recruiting type 2X fibers because I need power. No. I start out with type 1, and because I need more force, I then recruit the next and then the next. The idea I'm trying to get you to understand here is the order of recruitment doesn't change. It is the speed at which I switch between these that changes, okay? That's important to understand with this size principle in recruitment. Other notes that I want you to understand about motor units in general are that, let's just say this is a fascicle, right? You guys remember fascicles, a bundle of muscle fibers. And let's just say there's a whole bunch of these different types of muscle fibers inside this fascicle, which in reality is true. Is their distribution going to be type 1 on the left side of the fascicle, type 2s on the right side of the fascicle? No, that's not going to be the case. Why not? Because that's going to be producing biomechanically weird force vectors, okay? Uneven force vectors. So our contraction is going to be looking weird and not optimal. Instead, there is a homogeneous distribution of fiber types inside of a fascicle, okay? So the type 1s and the type 2s are intermingling between one another, creating what's known as this mosaic pattern, okay? So this mosaic pattern comes from the fact that there is a homogeneous distribution of muscle fiber types inside of a fascicle. And the concept I'm trying to demonstrate to you here is that, as you can see, one motor neuron can innervate multiple myofibers of the same kind, right? But it cannot innervate the same motor neuron can't innervate different types of myofibers. Similarly, one myofiber is only ever innervated by one type of motor neuron. So these myofibers are only ever going to be innervated by this motor neuron, okay? That's important. And finally, when we talk about motor units as a whole, you must understand that a motor unit, again, is the motor neuron and the muscle fibers. So when this guy fires, right, when this motor neuron gets recruited and it fires an action potential, it's not like only one of these myofibers that it's innervating gets activated. No, it's an all or none. When a motor unit is recruited, every single fiber in that motor unit must contract, okay? It's an all or none contraction in a motor unit after one action potential. That's important. Okay? All right. Next, a couple of questions to pose to yourself. Why is it that motor units exist instead of just having a one-to-one neuron to fiber? Why not just have one neuron innervating one fiber in every single fiber in a muscle? Why not do that? Well, it has to do with the structural efficiency. A one-to-one neuron to fiber ratio would require so many neurons, and that means that my spinal cord not only has to be bigger, but my brain would have to be bigger evolutionarily, and by proxy, the spinal canals and the skull would have to be bigger, and so this is just not optimal, okay? And if you're saying that, okay, well, let's not do a one-to-one neuron ratio, let's just have a singular neuron branch out and innervate every single fiber in the entire muscle. Right? So why not just have one motor neuron per entire muscle? And again, that's also not optimal because then we lose the ability to modulate force. A single neuron controlling every single fiber would create, again, an all or none phenomena where I cannot gradate my force like I would be able to before. And I wouldn't be able to, again, execute fine motor tasks. Every time I try to bring a water bottle to my mouth, I just smash it into my face because there's an all or none contraction. So the presence of these motor units is really, really beneficial to us because we get to do this force gradation, right? By doing orderly recruitment. We can modulate force output of a single muscle fiber. How? By just increasing action potential frequency, okay? So not only can I increase force production by recruiting more motor units, okay? So I have only one type 1 motor unit recruited right now. Let's say I draw another type 1, and if I recruit this guy, boom, now I have 2 type 1 motor units recruited, force output is doubled, okay? Instead of doing that, I could just increase the action potential frequency going down this one motor neuron to increase contracting of these guys. And we'll take a look at this at the end of the video. But again, this is the benefit of having these motor units. Okay, I can do this kind of stuff. Finally, the presence of motor units, this brings me to this point here. The presence of having motor units is important because I can participate in something called asynchronous motor unit recruitment. So what is it? Okay, well, let's take a scenario here. Let's take a look at your back muscles, your erector spinae muscles, for example, right? Those back muscles, they're not maximally contracting. In fact, they're submaximally contracting pretty much all the time to fight off against gravity and keep you posturally standing upwards, right? Or upright. Now, let's just say this. Is it one motor unit that is firing there? So if I have this graph here in time over seconds and force production, Is it just one motor unit that's going to fire continuously like this forever to keep your back muscle standing? No, of course not, right? Why not? Because this would fatigue the muscle, and guess what? If your muscle fatigues, then it's not going to contract, all right? And then you're not going to be able to keep your posture up anymore. So what is happening? Well, is it multiple motor units that are maximally contracting? Or is it multiple motor units that are submaximally firing to produce enough force to keep you up? The second option is a little bit more better, okay? So the way it works is you have, let's just take a simplified example. Let's say you have two motor units. You have motor unit 1 and motor unit 2. And in this case, motor unit 1 is smaller and motor unit 2 is larger, okay? Now, let's say I start out with motor unit 1. And so let's change the color to motor unit 1. And motor unit 1 starts off force production, right? And guess what? He produces a decent bit of force, but then in order to give him a little bit of break, what do? We shut him off. We shut this motor unit off so he can relax. And then we launch him back up again, and then we let him relax. Then we launch him back up, we let him relax. Launch him back up, let him relax. Why are we letting him relax? Because we want to give the fiber a bit of rest. We want to give the motor unit a bit of rest, right? If this was the only thing that was occurring, right, wouldn't your contractions be a bit jerky? Wouldn't your submaximal contractions be really jerky? Like when you stand up, wouldn't your back just constantly be shaking because of this like on and off and on and off and on and off motor units? It would. So why isn't it? And the reason why it's not doing that is because of this notion of asynchronous motor recruitment. Simultaneously as this guy is doing its thing, there's another motor unit, okay? And let me switch colors to this guy now. This other motor unit, though, it doesn't synchronously fire, okay? Look at it. So it doesn't synchronously fire. What do I mean by this? Well, take a look. What do you see here? There's a peak on this motor unit, and there's a valley on this motor unit. There's a peak on this motor unit, and there's a valley on this one. Peak here, valley here. Peak here, valley here. What am I getting at here? Whenever this motor unit is in a contracting state, this motor unit is relaxing. And whenever this motor unit is relaxing, this motor unit is contracting. So you see what's happening here, right? With this method, we can get multiple motor units to produce force and smooth out the otherwise jerky contraction that we would have from just this one motor unit having on and off and on and off. And the reason we can do that is because whenever this guy's off, this guy's on, okay? And whenever this guy's off, this guy's on. And so, again, if we plot the force production as a whole now, as a function of total force produced with these guys combined, you're going to get something like this. Total force is going to be this one continuous smooth force produced, all right? And that's the notion of asynchronous recruitment. It is for fatigue prevention, all right? Fatigue prevention. Now, this happens over hundreds of milliseconds, and you don't even notice it, okay? This is how fast this is occurring, okay? And again, it is to delay or prevent fatigue in a sustained contraction. And the example we were giving is, for example, the postural muscles, right? Because they have to have sustained contractions at all times. And the entire principle is just that the body alternates between motor unit activity, kind of like, shifts at a factory, right? And this gives the motor units that have been active an opportunity to rest while the other takes over. And this allows for a smooth production of force, not a jerky contraction. And please make sure to understand this. This only occurs during submaximal contractions, all right? And the reason is, during submaximal contractions, only some of the motor units must maintain a desired level of force over a period of time. But in a maximal contraction, all muscle fibers must participate. And so it is impossible to kind of just jump between one and the other because they're all contracting as maximally as they can. All right? Great. So now that we've finished here, let's go ahead and take a brief look at this chart. Now, this chart right here is going to cover a lot of the stuff we talked about there. And so this is going to be a lot of review for us. So type 1 fibers, what is their recruitment order? Well, type 1 is easiest to recruit. Type 2A would be hard to recruit. And then type 2X would be the hardest to recruit. In fact, during regular bouts of exercise, type 2X is often very frequently recruited. And so hypertrophying these fiber types is really hard because recruiting them is really hard. Glycolytic enzymes, remember the name: slow oxidative, fast oxidative glycolytic, and fast glycolytic. The name tells you what you need to know. The glycolytic enzyme concentrations in a slow oxidative fiber are probably going to be not that high. Why? Because its main method of fuel is oxidative phosphorylation. So low. Type 2A, high. And then type 2X, highest. Okay? What about the force production? Well, again, that has to do with the diameter of the fiber, right? Fiber type diameter is bigger in a type 2X, so we will have the highest force production. Why? Because it can stack more myofibrils. We already talked about this. So low, high, and highest. Okay? What about the mitochondrial concentration? Well, the mitochondrial concentration, type 1 fibers are fatigue resistant and they're slow oxidative. So you would expect their mitochondrial concentrations to be mighty high. And it is. Type 2A, a little bit different, right? They're in the middle, fast oxidative and glycolytic. So they're going to be medium amounts. All right, and this guy's obviously going to be the lowest. What about the contraction and relaxation time? So remember, we took a look at those contractile kinetics, right? Those graphs that looked like this, this, and then this, right? So if we look at the contraction and relaxation time, the type 1 fibers are the slowest. So they contract slow and they relax slow, all right? The type 2A fibers, they contract fast and they relax fast. Why? Because they are fast twitch fibers, right? This guy is slow twitch. The type 2X will have the fastest twitch time, and we talked about that, right? Yeah, we talked about that there. What about the capillary to fiber ratio? So capillary to fiber ratio is an interesting one. So if you look under the microscope, right, and you see one fiber here, and then you got another fiber, another fiber, another fiber, another fiber, you can physically count the number of capillaries that are surrounding that fiber, and then you make a ratio out of it, okay? And so this capillary to fiber ratio is useful in scenarios where we can study exercise adaptations, because then we'll have capillary growth or angiogenesis. Either way, type 1 fibers, think about this. What would their capillary fiber ratio be, high or low? Well, they are slow oxidative. They have a lot of mitochondria, which means they require lots of oxygen. Oxygen comes from the blood, so they require a high capillary to fiber ratio. And that's true, okay? So high capillary to fiber ratio. Type 2A. Type 2A, they're in the middle, so they have to be medium. And this guy, type 2X, has a low capillary to fiber ratio. Now, does this mean that these capillaries are absent? No, it's just that relative to the type 1s, it is low, okay? These values are all relative to one another. What about fatigue resistance? Well, fatigue resistance, again, this guy relies heavily on oxidative phosphorylation. So his fatigue resistance is going to be pretty high. We took a look at those fatigue curves earlier, right? Type 2A, also medium. And then type 2X, of course, the lowest fatigue resistant. Okay. A couple other ones that I added in here because I thought they would be important is myosin ATPase activity. So remember the myosin had, right? It had the two binding sites, the actin binding site and then the ATP binding site. And within the ATP binding site, there's a myosin ATPase, right? That enzyme has different isoforms. Isoforms are kind of like, you can think of it like this. You know how you have ice cream, right? And you have different flavors of ice cream, right? You got chocolate, you got vanilla, you got mint chocolate chip. They're all ice cream. It's just they're a little bit of a different flavor of ice cream. That's kind of like what an isoform is, but for proteins. So they're the same protein. They do the exact same function, or they're the same enzyme, they do the same function, but they just function a little bit differently, okay? In this case, the myosin ATPase is a little bit faster, okay, in type 2X. So the myosin ATPase activity is faster. Now, this ATPase activity has implications for cleavage of ATP, so how fast I can cleave ATP and recock the myosin head into an energized position. And it has implications for cross-bridge cycling, and the affinity that ATP has here is different between these different fiber types based on their myosin ATPase. Okay, so that's important. Type 2A, well, its myosin ATPase is medium, and this one is slow. Okay, and this should make sense to you. Myosin ATPase activity, right, is the thing, right? It is the cleavage of ATP that actually allows for the energization of the myosin head, right? And for, the binding of ATP to occur, it is to allow this myosin head to detach from actin, then it gets hydrolyzed, right, into an ADP and energetic phosphate, then the head is energized, and now we have formed a new cross bridge and we're ready to go if calcium's present. So that's important, because the faster that occurs, the faster the cross-bridge cycle occurs, and the slower that occurs, the slower the cross-bridge cycle occurs, okay? The next one is sarcoplasmic reticulum development. So a type 1 fiber, it doesn't really need to produce a lot of force. We've established that already. And morphologically, its sarcoplasmic reticulum is not as developed, okay? So it's poor development compared to the type 2X. It has a lot of SR development, okay? There's a lot of branching. It's a very giant sarcoplasmic reticulum. And so that's important because they need a lot of calcium. Why? Because calcium is the determinant of force production. We talked about this in the previous video. And so if they have calcium, then they produce a lot of force, which is, of course, what type 2X require. And so this guy would be medium SR development. The last thing I added in here is myoglobin concentration. And if you don't know, myoglobin is a molecule similar to hemoglobin, but instead of being in the blood, is in the muscle, and it doesn't have multiple polypeptide chains. It only has one. And it essentially binds oxygen and kind of stores oxygen and delivers it to the mitochondria for use later. And so based on everything we know about these fibers, which fiber type do you think would have the highest myoglobin content? Well, it would stand to reason the one that has the highest has the highest oxygen use would have the highest myoglobin content, and that would be the type 1 fibers because they're oxidative phosphorylation, right? So high, of course, medium, and then low. Okay. All right. Now let's talk a little bit about the color, because I said I'd tell you why these guys are slow twitch red, why these guys are fast twitch red, and why these guys are kind of pale white. Okay. The color, there could be many reasons for it, and it's not likely just one thing. It's probably just a combination of all of them, but I'll tell you all of them. So one is the mitochondria, the other is the capillary to fiber ratio, and then the other one is myoglobin content. So these three things will contribute towards the redness of the fiber. Why might you say that? Well, capillary content, if the capillary content is high, well, what's in the capillaries? Blood. What color is blood? Red, right? So that's going to make the fiber appear more red. But you might be like, okay, well, what if the meat is dead? What then? There's no blood going to it, so why does it still appear red? The other examples are going to be mitochondria and myoglobin levels. Both myoglobin and mitochondria contain molecules of heme. In fact, heme synthesis occurs in the mitochondria. And so heme is a pigment, right? It's found in hemoglobin. That's why red blood cells are red. And so it is the combination of the mitochondrial heme and the myoglobin heme content that makes these fibers look red. And the absence of them makes the rest of these fibers look less red, okay? And, of course, type 2X have low amounts of all three of those factors, and so they will be white in appearance, or pale pink, if you will, okay? Last note about myoglobin is that whenever you grab like this, juicy steak and you've cooked it on, wherever you're cooking it, and push down on it and this red liquid comes out, that's not blood, right? That's myoglobin, okay? And it's red because of this exact reason we talked about, because of the heme inside of it, okay? So that's all the myoglobin coming out. All right. So we talked a lot on this page, okay? But now let's go ahead and talk a little bit more at the cellular and molecular level about force production, okay? So this page was really just about getting you to understand motor units, the different types of motor units, small and large, right? The different types of muscle fibers, the characteristics of motor units, asynchronous recruitment, right, for fatigue prevention, and the Henneman size principle. Now we're going to talk about force production. How might we increase force production, voluntarily? And we've talked about this already. And additionally, how does a single muscle fiber reach maximal contraction, okay? So let's go ahead and talk about that. So we've already looked at this graph in the previous video, right? We said that the skeletal muscle's resting muscle membrane is minus 90, and it reaches a peak of positive 30, right? And we said this is the action potential. It lasts 1 to 2 milliseconds or 1 to 5 milliseconds, like I said, depending on who you ask, right? And it's this portion. We said that as the action potential is occurring, you can see that there's no force being produced, and that's because this is the latent period. And we said that the latent period is all of the events of the action potential starting at the sarcolemma and making its way down the T-tubules, activating the dihydropyridine and the ryanodine interactions, allowing for calcium to be released from the SR, calcium making its way and binding to troponin C, moving the tropomyosin, and finally, start of cross bridges. That's the latent period. That's how long it takes. It's about the same time as this, okay? I don't really have an exact time for it. I couldn't find an exact time, but it's about the same. It's about the same. Like, it's a little longer, as you can see, okay? So this is 1 millisecond. Maybe this is like 5, okay? All right. Now, again, this is a graph for just a general fiber. We've already talked about the twitch kinetics of different fiber types, but a general fiber, they usually teach it as 100 milliseconds, right? But we know that, for example, this is more representative of a type 1 fiber curve, whereas a type 2X fiber would be around 20 to 40 milliseconds, okay? So we've already talked about this. Of course, the graph has a rising phase, the contraction phase, and then it has a relaxation phase, right? And again, we spoke about the fact that the relaxation phase is really just the uptake of calcium, right? By the circuit pump, and the contraction phase is just a release of calcium, okay? And so you should already be ringing some bells in your head that calcium availability is the thing that's actually going to regulate muscle contraction, okay? And the force production even. And again, I think we talked about this in the previous video. Now, the other thing that we were mentioning was that, and we didn't build on this a lot in the last video, was we can overload the circuit pump so that it can't keep up with the amount of calcium that is inside of the cytosol. And what that's going to do is it's going to saturate every single sarcomere, or more so every single troponin on every single sarcomere, so that force production can reach its maximum amount. And we said the way to do that, the way to release more calcium, is to launch more action potentials, right? So we said that we launch more action potentials, right? We can release calcium. And there's a reason why we can do that. The reason we can do this is because the action potential has a very short duration. And so its refractory period will already have been done by the time I want to launch another action potential. And another thing is that, write down short duration action potential. The other thing is that the twitch duration itself, okay, is so long, so it's around 100 milliseconds on average, right? It's so long that I can squeeze in a whole bunch more action potentials, right? Because they last 1 millisecond. So if I wanted to, I can squeeze in around 100 or maybe less, like 80 hertz. Okay, and hertz just means action potentials per second. So I can squeeze in a whole bunch in there. Okay, or sorry, maybe not 80 hertz because this is in milliseconds. Okay, but either way, I can squeeze in a whole bunch by the time this fiber relaxes. And you know what that's going to do, right? That's going to increase force production, right? So the muscle is relaxing because there's uptake of calcium. But if I launch another action potential and another action potential, what's going to happen? Well, we're overloading this circuit pump. So I'm going to get a graph like this now, right? It's going to go up and it's going to keep going up. And we're going to take a look at that. That right there is a concept of twitch summation. And we'll talk about it in great depth in just a second. But first things first, let's go through how we can increase force, okay? There are many ways for us to increase force. Some of them are physiologically relevant to us, meaning that we can actually do something about it. The other ones, we can't really do too much about it, okay? So let's write a note here for ourselves. How do we increase force, okay? Be sure to understand that force is not the same thing as power. We'll take a look at power later in a graph, but again, this is not the same as power. Power is force times velocity, all right? And velocity is distance traveled over time, okay? All right, so we'll write that distance over time. Okay, one way that, we already know of increasing force is what? Well, we have multiple motor units in a skeletal muscle, right? And so we were talking about it right here with the orderly recruitment. If I can't match the force that I need to maintain an exercise intensity level, then what do? I just switch to the next myofiber. Well, how do I do that? I do that by recruiting the next motor unit, right? So a general answer we can give here is by recruiting more motor units. Okay, recruit more motor units. And what does this affect, guys? This affects the number of either the same or different types of fibers being recruited. Okay? All right. The next thing we can do is instead of just recruiting more motor units, I can just increase the firing frequency of that one motor unit. Okay? So increase firing frequency of motor units. All right? And what this does, and this is what we're about to look at, is it affects the individual force production from muscle fibers being innervated by that motor unit. Okay, so if we go back here, guys, if we go back to this diagram, here's my motor neuron. Here's the fibers it innervates. If I want to increase force production from this guy, I can only thing I can do here is send more action potentials down to overload the circuit pump, right, on these muscle fibers. And that's going to affect this muscle and this muscle's individual ability to generate force. Okay, because remember we said that when a motor neuron fires, all of the myofibers it innervates are going to contract, okay? And so the way we can affect the force production of these myofibers is by launching more action potentials, all right, in this motor neuron. And so that would be written as increasing firing frequency of a motor unit. What does this look like? Let's take a look at it on this graph. So if I have a single myofiber force over time, okay, let's just say it's just some unknown unit of time, right? And I take this muscle fiber, right, and I'm going to make it do a couple of twitches, okay? So I'm going to depict my action potentials here with arrows. So let's say I draw one arrow here, and that's an action potential. And I would like you to associate this action potentials with effort, okay? Effort. If I want to lift a book, guys, does that take a lot of effort? No, it doesn't take a lot of effort. So do I need to send many action potentials down the axons of my motor neuron to innervate my myofibers and cause them to contract more forcefully? No. But if I were to try to lift a table now or a desk, then things would be different. I would have to launch more action potentials. I would have to put more effort into doing that task, okay? So let's say I launch one action potential here. And of course, after the latent period, I get one twitch. This is a single twitch, the contraction phase, the relaxation phase. Let's say I launch another action potential and I get a second twitch. But let's just say as this is coming down, before it has fully relaxed, I launch another action potential. Okay, I launch another action potential. And what is that going to do? What that's going to do is it's going to cause a release of calcium, more calcium from the SR. And so when we were already uptaking calcium here, and now I just release that same calcium back into the cytosol, that has just negated what I've done. And guess what? That's going to increase force production. Okay. Now, let's say I keep going. Let's say I don't stop here. Let's say I launch another action potential as it's already in its rising phase. Then what's going to happen? Well, it'll keep going and it'll go higher. Okay. And this process of me summing the tension or force provided by the muscle fiber together is called twitch summation. because I'm adding up the force production from the previous twitch to the now added calcium, and more cross bridges forming and an additional twitch occurring, okay? So now this is where the diagram's gonna start to look messy. What if I just keep blasting this cell, this myofiber, with a whole bunch of these action potentials? And I just start increasing the frequency now. They're gonna start happening more frequently and frequently and frequently. Eventually, guys, what's gonna happen? Eventually, look, I'm gonna get these kind of wave-like upwards, and then eventually, it's gonna become this continuous smooth line right over here. And if I keep going, right, if I increase the action potential frequency even more now, okay, so now they're happening more frequently than they've ever been happening before. Notice what's happening. The line stays there. It doesn't go down, but it doesn't go up either. What does that tell you? That means that I have reached the maximal amount of force that this singular muscle fiber is capable of producing. What is that called? We have a term for that. That is called tetanus, okay? Or a tetanic contraction. It has nothing to do with the disease tetanus, okay? That is caused by Clostridium tetani, an anaerobic bacteria that makes the motor neurons that sit in your spinal cord hyperexcitable by blocking central nervous system inhibition, okay? Completely different from this. This is just a term we happen to give when a singular muscle fiber, after repeated stimulation via action potentials and release of calcium in the cytoplasm, reaching its maximal force production, okay? Notice the action potential frequency has gone up. The force production hasn't gone up though. Why? Because this is the maximum amount of force this fiber can produce. Now let's do another experiment. Let's say that this example that we just did was actually of a type 1 fiber. Now let's take a look at how a tetanus would look like for a type 2 fiber. Okay, so let's say we go with the same action potential. I launch an action potential, and after a while, there you go, it does a twitch. What do you notice about this twitch? It's thinner, okay? The contraction and relaxation time is higher, and the force production of a single twitch is higher, okay? We've learned about this already. What's next? I launch another twitch. Boom. And then I launch another action potential here. I should say action potential, not twitch here. I launch another action potential here. And it goes, and as soon as it's about to come down, I launch another action potential, and boom, it adds on top. All right, and then another one, all right, and it adds on top. And it starts to come down again, and another one, okay? Starts to come down, another one, and another one, and another one, and it keeps going, right? And it's going to go, and until it reaches its own tetanic contraction, okay? Now, this right here is tetanus, but for a type 2 fiber, okay? Now, we follow the same action potential frequency pattern here. In reality, this wouldn't occur, right? The different motor neurons for the different fiber types have their own firing frequencies. So the type 1 fibers are more tonic, okay? They have more of like a basal firing rate, whereas the type 2 fibers, they have more like bursts of large impulses and then nothing for a while, and then bursts of large impulse again, okay? They could be somewhere around 50 to 100 hertz in those bursts, whereas I believe a type 1 should be somewhere around 20 hertz and even less, okay? So this diagram of a tetanic contraction, what does it tell you? It tells me that if I increase my motor neuron firing frequency, then I am going to increase force production at the myofiber level. And so one way that I can actually increase force, all right, is either by recruiting more motor units or by increasing the firing frequency of motor units, okay? And this is this, all right? These two are what you can control. You have voluntary control of this, okay? This is central nervous system control of your actual force production. Why do I say central nervous system control? Because we have to come back here. It is your brain, aka you, that sends a signal from the central nervous system down to the spinal cord to make this guy activated, okay? He's not going to fire on his own unless you're actually infected with tetani, right, or tetanus. So this is the two ways you can actually change force production, all right? Now, there is an important concept to understand here. We understand that one twitch is never enough to have any effect at the whole muscle level, literally nothing. But what we do have to understand is that one tetanus coming from one fiber is also not enough for the whole muscle level. It would take multiple fibers going through this tetanic contraction to make a difference, okay? And when we say increase of force production, please understand these two don't occur separately. They're not mutually exclusive. They will occur more often than not together. Because again, if you equate action potentials to effort, right? As I increase my effort, I'm going to first most likely increase the firing frequency of my motor unit, and then I'm going to be able to recruit more motor units later if I can't meet the desired force production just off of increasing the force frequency of my current motor unit, okay? This tetanus right here, it's not going to be sustained forever, okay? So what I mean here is that if the action potential firing frequency stops, or it falls below the threshold to keep it at this plateau, then you start going down, okay? It's either that or the muscle hits fatigue, okay? And this can be from multiple different areas, okay? Muscle fibers can hit fatigue in multiple different ways. We'll probably have a video discussing that later, okay? But there's central nervous system levels of fatigue, neuromuscular junction fatigue, myofibril level fatigue, a whole bunch of stuff, okay? All right, so now that you understand this, we can now dig a little bit more in different ways of increasing force production, okay? But please understand, these are the ones that you can modulate, and these are the ones that are really important. The other ones, you don't really get a say in them, okay? That's just how it is, all right? That really has to do more with the muscle mechanics rather than, you having to modulate it, okay? And an example of this would be muscle length, okay? There's this thing called optimal muscle length. It's represented by this symbol, optimal muscle length. And we could draw a graph of it. So I'll go ahead and draw the graph in for you guys. On the x-axis, we have the percentage length of muscle fiber from resting length. So if resting length is 100%, right, that already correlates to the highest force production. That right there is this value. That's the optimal length. So I'll go ahead and sketch that in for you. This is the optimal length. And at the optimal length, the muscle, this is the resting length of the muscle fiber already is at its highest force production. At this point, if we were to draw a sarcomere, okay, there is equal and the most optimal overlap between the myosin cross bridges, between the myosins and the thin filaments. So there's a lot of cross bridges formed here. This is the optimal length. And, in the body, the resting muscle length is when the muscle's not actively contracting or passively stretched. This is already the case, okay? This is already the case. So for example, optimal muscle length in like the biceps would be like 90 degrees, okay? Now, when you stretch the muscle fiber in either direction, okay, so when you shorten it or you stretch it, okay, so this would be shorten, right? So we're moving away from 100, which is the resting muscle length, which means we're shortening the muscle fibers, and over here we're actually lengthening the muscle fibers. And again, remember, these are percentage values, 100%, okay? The normal physiological ranges, okay, meaning these are the actual, this is as far as we can stretch in a human body, are this. This is the length. Do you notice that there's not a lot of drop off in force production here? Okay. And like I said, you can't really do anything with this muscle length. And why is it that this is the actual, curve for humans? Well, it's because there are restrictions imposed by skeletal attachments, and muscles cannot really go above 30% of their length in either direction, whether it's stretching or lengthening. And it just has to do with the skeletal attachments, okay? So, for example, I cannot hyperextend my elbow. I mean, I could, but that would be painful, right? So there's literally a limit to how far I can stretch my biceps muscle. And you can see at the outer limits of these ranges, the muscles still achieve majority of their force production, okay? Now, it is not shown here, but for example, this is 100% force production. At either end of those ranges, you'd be around 50%, okay? So at either end, the muscle fiber, I should say, is still giving around 50%, all right? It's not bad. Where do these other ones come from? Well, these come from actual, research studies where they've taken the fiber, they've isolated it, and now they're stretching it beyond the point of whatever. And for example, like I said, the myofiber here, the sarcomeres would have the best arrangement possible, right? Over here, and over here and over here, okay? It's just was more extreme. But in both of those scenarios, the percentage of maximal contraction that can occur there is going to be decreased. And that's because there is, the fiber itself is stretched, so the sarcomere is going to be really stretched. And if the sarcomere is really stretched, you can imagine this is my thick filament, right? And now these are the thin filaments. Look at how far the thin filaments are from the thick filaments, okay? And so this thick filament and this thick filament, they're not going to have any thin filament to interact with. So that means less cross bridge, less force production, okay? On the other end of the spectrum, okay, so let's just say around here, something different is occurring, okay? In this case, I am actually shortening the muscle. And so what's going to end up happening is, again, here's my thick filament, okay? Here is my Z line, okay? And now the thin filaments are going to end up overlapping with one another. Again, this doesn't occur in physiological situations normally. And when they overlap, of course, there's less cross bridges available to be made, okay? And the other thing is that the thick filaments are kind of being forced against this Z line, okay? So there's less myosin heads being able to actually interact with those thin filaments as well. So there's a couple of multifactorial stuff going on there, but that's a optimal muscle length concept and how that affects force production, okay? I'm sure you've noticed that you're stronger at, for example, holding a dumbbell on your hand when your biceps is at a 90-degree angle than if you were to just hold a very heavy, that same heavy dumbbell and try to hold it at the bottom of a contraction for your biceps when it's super stretched, okay? So that gets harder. One question we did not end up addressing here, which I'm surprised I forgot about this, is for tetanus. What determines whether tetanus occurs or not? And you should already know the answer here. It's calcium, right? It's the duration of calcium availability in the cytosol, okay? If more calcium is available, then, okay, we're going to be able to reach tetanus. If less calcium is available, then the muscle's going to start relaxing, okay? That's just a given, okay? All right, now let's get back to this. So now we know 3 ways muscle force can be increased. The other one is, it has to do with the velocity of contraction, okay? So the higher I should say this, the lower the velocity of contraction, the higher the force production. But there is a nuance. There's a nuance here, okay? Which we're probably not going to get to in this video, all right? But if we were to draw the force velocity graph, which looks like this, here's force, and here's velocity, right? Going this way increases velocity, going up increases force. Great. The graph looks like this. What does that tell you? Well, this is showing me that as the velocity of the contraction is increasing, the force that is produced is decreasing, as it's seen here. But if I read it the other way, as the velocity decreases, the force starts to increase in this nonlinear fashion. Okay? That's the force velocity curve. And of course, you eventually reach a point where you have this, and that's oftentimes called the unloaded shortening velocity. Okay? Unloaded shortening velocity. Okay? And you don't really have too much to worry about with this. Okay, this is more of like a lab concept than it is something that actually occurs in humans. What I do want you to understand, though, is that this graph is a little misleading, is that it's only telling a part of the story. So we'll talk at the end of this video about the kinds of contractions we can have. Actually, you know what? Well, let's talk about it right now, and then we'll get back to this. So we have different kinds of contractions, right? We can have isometric, isotonic, and isokinetic. We're not going to talk about isokinetic because isokinetic just means contraction occurring with the same velocity. Isotonic, though, refers to the same force, okay? Isotonic contraction means that the tonic or tension, it remains the same, okay? Isometric means that the muscle is contracting, but it's not changing in length. It's not shortening. So this right here would be, for example, an isometric. This would be well, these two are isotonic, but they're different examples of isotonic. So this is a eccentric contraction, and this is a concentric contraction. Because in both isokinetic and isotonic, the length of the muscle changes. But in isotonic over here, we see that we have an eccentric contraction, right, which is when the muscle is stretched, and the concentric is when the muscle is lengthened. Sorry, not lengthened. This is lengthened. Over here, it's shortened. Okay. Now, how does this occur? What is the thing that's making this occur? It has to do with muscle tension and muscle load. Muscle tension is just force. So, muscle tension. And this guy is muscle load. So over here, you can clearly tell this is a elbow here, and this is your biceps, right? And this is a load that you are holding. So generally speaking, when the muscle tension is equal to muscle load, you have an isometric contraction occurring. So 10 kilograms of, let's say my muscle is producing 10 kilograms of force, and my muscle load is 10 kilograms of force. Then in that case, these two equal out, and so there's an isometric contraction. Now, in a scenario where the muscle load is greater than the muscle tension, then an eccentric contraction occurs. So let's say this is 20 kilograms now, and my muscle is still producing 10 kilograms of force, then the load is greater than the tension being produced, and so that's going to end up stretching the muscle. Do not make a mistake here. These eccentric contractions, the sarcomere is still going through a contraction. The sliding filament theory is still occurring. It's just now they are attempting to slow down this descent, meaning they're trying to have an controlled eccentric contraction occur rather than just letting the weight fall down to the floor, okay? And so they are still contracting. It's just they're contracting eccentrically. In a concentric contraction, this is more easy to visualize for people. This is where the load is less than the actual force produced. So the muscle tension is 30 kilograms here, whereas the weight, the load in my hand is 10 kilograms. And so in this case, my myofibers and the fibrils inside them can actually overcome this tension, this load, and cause concentric or shortening of the muscle. So these are the three different types of contractions we have. Now we know this. Let's come back to this force velocity curve, because this force velocity curve is important. This portion of the curve is for a concentric contraction. So from about here all the way down to here is about concentric contraction. And I'm sure you've experienced this. If not, go and grab something right now. Grab a book and lift it as fast as you possibly can. All right? The force production there is going to be very low. Why? Because you used a whole bunch of speed, a lot of momentum to get that weight up. But if you grab that, well, perhaps not a book, but if you increase the weight and you grab something like, I don't know, 10-pound dumbbell, and you try and lift that real slowly, it gets a little harder, okay? It gets a little harder. And so force production there is higher because the velocity is lower, okay? Now, like I said, this is concentric. The point at which velocity is 0 is an isometric contraction. So this right here is isometric. And in this specific graph that I've drawn, an isometric contraction produces the highest amount of force, okay? Now, why? Because the velocity is 0. So again, isometric means that the length of the muscle is not changing. If the length of the muscle is not changing, that means there's no movement occurring. And if there's no movement occurring, the velocity is 0, right? Velocity is distance over time. If there's no distance traveled over a given period of time of the muscle, then the velocity is 0. But contraction is occurring, okay? Take your hand right now and just squeeze it into a fist and just squeeze as hard as you can. That's an isometric contraction, okay? Go and push a wall as hard as you can. Your muscle's not shortening, but it's contracting, right? So isometric, this is the point of the graph. This is isometric contraction because the velocity is 0, highest force production. You'll see later that, in maybe other courses or videos that this is not always the case. This graph isn't actually complete. This graph needs to be dragged on this way. And this portion would be the eccentric portion of the graph. And you'll see that if the velocity is decreasing in the opposite direction, all right, so if I'm bringing the weight up concentrically, this is the velocity increasing in a positive direction on this x-axis. But if I bring the weight down, the velocity would be increasing in the opposite direction. And you'll see that it's actually the reverse. The higher the velocity is at that point, the higher the force production, for reasons we're not going to get into in this video, okay? Just wanted to mention that so you have a complete info. Now, sometimes with this graph, you might see something like this. And this is really describing the math. Remember over here I said power is force times velocity? So I got force here and I got velocity here. So I can plot power on this graph. And this is what this is. You can see that the power starts to increase, right, as I reach this optimum velocity. You know what they call this? They call this P optimal, okay, for optimal power. The maximum amount of power that can be produced is at V optimal, so the optimal velocity, and the optimal force production, okay? You don't really know what this is until you experimentally measure it. So again, that's why I'm saying that the two main methods that you have of just increasing force yourself is these. The rest of these are just kind of, biomechanically there, okay? All right. We got a couple more, I promise, and then we're done. This one is the extent of fatigue. We're not going to talk about this, but this should make sense to you that if fatigue is onsetting on a muscle fiber, then guess what? That muscle fiber is not going to be able to contract anymore, okay? The next one is the thickness of that fiber, okay? So that comes back to the fiber type, okay? Different fibers, different thickness. And so that has to do with the number of myofibrils or sarcomeres inside of that fiber. And this can actually be changed by training, right? You heard of hypertrophy? That is increasing the diameter or the thickness of the muscle fiber, stacking in more myofibrils in there, all right? And so this is another way of increasing force. So perhaps there are three ways that you have of voluntarily increasing force, and one is these neural ways, and the other is by training and getting this adaptation of hypertrophy, where now you have higher force production just off of the fact that you have more myofibrils or a thicker myofiber. Okay, and again, we've talked about this with the different fiber types, right? So that's all the force production stuff out of the way. Last topic that I want to touch on is going to be fiber type distribution. One thing you should all be familiar with is that, and I've already mentioned this before in previous videos, that muscle fibers are terminally differentiated muscles. They are out of the cell cycle. They do not go through mitosis. The only way they regenerate is via the satellite cells that they have present on their surface, okay? And so hypertrophy of a muscle cell means that the muscle cell gets bigger. Hyperplasia is when the muscle increases in number, okay? You actually get more muscle fibers. That is really rare. That doesn't occur much in humans with training. Hypertrophy is what we see, okay? So why does this matter? This matters because genetics come into play. Whenever you're born, you have your number of fibers set, okay? So what I mean here is that the total number of fibers per muscle is fixed at birth for everyone. But individuals can be born with different total number of fibers per muscle. Okay? And this is where the discussion of an elite athlete comes in. All right? Elite athletes describe individuals who are excelling at the height of their athletic career. Okay? If I take a muscle biopsy from these guys' muscle fiber, I would expect to see some different fiber type distributions. And a great example that's always taught very well is comparing a sprinter, a marathoner, and a non-athlete. So somebody like me or you watching the video, unless you're, an elite athlete. These comparisons are going to be made in a single muscle fiber. Sorry, in a single muscle. And then we're going to examine the different fiber types. If I take a sprinter's gastrocnemius, the calf muscle, the marathoner's gastrocnemius, and a non-athlete's gastrocnemius, and then I do a muscle biopsy and I start counting on the muscle fibers in them, right? And I do a percentage out of total, okay? So I say, okay, this guy had 3,000 muscle fibers, and in fact, a gastrocnemius actually has around 1 million muscle fibers in it. And so I count out, of that 1 million, what is the percentage that is a type 1 fiber, what is the percentage type 2A, and what is the percentage that type 2X? What would you expect to see? Well, a sprinter, somebody who does like a 100-meter dash, right, at the Olympics level, for example, you would expect them to have high amounts of power-producing muscle fibers. And that's correct. So I would expect them to have more type 2X, type 2As, and very little type 1s, okay? And the actual percentage, the average out values for multiple studies is around 25 to 30% type 1, and type 2s just grouped together is going to be around 70 to 75%, okay? And so this represents the percentage of fibers in this guy, this sprint, or this individual's gastrocnemius muscle, right? That is type 2. The 25 to 30% is the percentage of this individual's gastrocnemius that is type 1 fibers, right? If you look at a marathoner, again, elite marathoner, this dude, what would his gastroc look like? What I would expect, a lot of fatigue-resistant muscle fibers, all right? Because this guy doesn't need force production. He needs to maintain a constant level of force production at a long period of time. And we looked at those fatigue curves before. Type 1 fiber was the one that's excelled at it, right? At 60 minutes, it still had 100% of its maximal force production, right? So this guy would tend to have the exact opposite phenotype, okay? And the exact values for this are going to be around 70 to 80%. And type 1 and type 2, again, joined together, are going to be 20 to 30%. This makes sense. In a non-athlete, though, guess what? This might blow some people's mind. In a non-athlete, it's pretty much a 50-50. Pretty much a 50-50. And you might be saying, well, hold on now. This doesn't make sense. The gastrocnemius is supposed to help you for walking. Why isn't it that I don't have a, more distribution of type 1 fibers than I have this? And that's because it has to do with your genetics, okay? Muscle fiber type is mainly genetic. If you happen to have in your gastroc, in your, whatever muscles that are used in running, okay? There's a lot of them, but let's just say gastroc because that's a big one. If you were born with that, if this is specific fiber type composition, you would know it. And the reason I say you would know it is because you would start out playing some form of a sport at high school level, at college level, and you just are better than most people without training. And you're just excelling real good. And this is going to get you to those high levels of competition. This is going to allow you to excel. Now, does this mean that training is not necessary and you don't need to train anymore because you're just that guy and genetically gifted? No, absolutely not. There are many different adaptations that come from exercise and training specifically that genetics is not going to get you. However, the one thing that is genetically determined here is going to be these fiber types, and there's nothing you can do about it, all right? In a non-athlete, like I said, it's pretty even. It's around 47 to 53% type 1s and 47 to 53% type 2s. So comparing these three should give you this good idea here, all right? Does this mean that the non-athlete cannot change its fiber type distribution via training? Does this mean that the non-athlete can't get benefits from training? Absolutely not. They can. And in fact, training does cause fiber type transitions, but it is very, very specific. So interconversion of the fiber type occurs only between type 2X to type 2A. So it doesn't matter if you train endurance. It doesn't matter if you train resistance. Study after study after study has multiple times proven that this shift occurs. Type 2X fibers will shift to type 2A. And again, it really does have to do with the fact that it's so hard to recruit type 2Xs. And the metabolic profile of this is just much more favorable for recruiting. That's one of the reasons. There's other reasons, of course. But this type 2X to type 2A fiber type conversion does occur. Okay? And so you will get more of these type 2A fibers via training for long periods of time. What doesn't occur is type 2X or type 2A transition to type 1. This doesn't occur in humans. It doesn't occur in humans. There are models of rat models that you can do to initiate this kind of process. It is called chronic contractile activity. It is where you attach a electrode with a battery pack towards, for example, a rat's gastroc, and you leave it to contract at a very specific frequency, around 10 to 20 hertz, for example, and you leave it to contract 24 hours for multiple weeks on end. And you will, after taking that muscle and biopsying and getting a sample and doing histochemical analysis, you will see that this shift has occurred. But this is not possible to do in humans because we just cannot provide that level of stimulus continuously. So theoretically, it's possible in humans, but in practice, maybe not. All right. And a couple of other notes that I do think it's important for you to understand is that, like I said, new fiber growth or hyperplasia with training is possible theoretically, but it's really rare in humans. The fibers do grow in diameter, though. They hypertrophy. And this is a really important distinction. When they hypertrophy, they constitute a higher percentage mass of the whole muscle. Okay? It is not like your percentage of muscle fibers is increasing. It is the percent mass is increasing relative to the whole muscle. And again, it's really important to understand this biological variability in fiber type distribution exists. And one more thing is biological variability in different fiber types in the same human exists. So we were talking about gastroc here having 47 and 53%, but if you were to look at your lumbricals, which are your hand muscles, right? They, instead of having, like the gastroc, instead of having 1 million fibers in them, they only got a couple thousand fibers, okay? Maybe around like 4,000 or 2,000. And so just off of the virtue that they have less fibers, they're going to have a smaller percentage of, for example, type 2 or type 1 compared to the gastroc, because the gastroc just has more fibers. And sorry, not percentage, but numbers. But if you were to compare percentage-wise, a muscle fiber or a muscle like the lumbricals, they don't need a lot of type 2Xs. So if you were to look at that fiber, right, or that muscle, they might have more type 1 fibers than they have type 2, okay? Or your extraocular muscles, they might have more type 1 than type 2. But in a lot of these other muscles, there is a big mixture going on, okay? And another important fact to understand is that if I were to total out every single one of your fibers in every single one of your muscles, I won't land at a 50-50 distribution of a type 1 and type 2, okay? That's just not how it works, all right? And one last thing is that at the elite level, there seems to be no major sex differences in fiber type distributions, okay, at the elite level, all right? And so that is just a cool factoid for you to have. There are exercise physiological differences for sure, but fiber type distribution doesn't seem to be one of them, okay? All right, guys, that's going to finish it for this video. If you stuck around, thank you so much. It's a long one. We had to cover a lot. I hope it made sense. I hope it helped you. And if it did, leave a comment, like, subscribe, and I'll see you in the next videos.